Good morning, everyone. Or actually, good morning, Miriam and Sharat. Thank you for coming. I'm Molly Starback, Director of the Office of Postdoctoral Services, and we are very pleased to have Debbie Swain-Lentz with us today. Debbie is a former postdoc. When were you a postdoc? Like
2016 to 2020.
2020. Okay. And Debbie is now the director of the Duke Sequencing and Genomic Technologies Core Lab, which I feel like is the most popular one. Is that me? I don't want to make it a competition, but I feel like
we are heavily utilized.
you're heavily utilized. Right. All right. So thank you so much, Debbie, and please take it away.
Yes, of course. So thank you for having me. As Molly said, I am the director of the core facility for sequencing and genomic technologies. Essentially, if you have questions or experiments about genomic technologies, such as DNA sequencing or RNA sequencing, you can come to us. So we offer a wide range of services for your experiment, and that includes library prep with these two staff members right here, as well as bioinformatic analyses shown by our collaborator Alejo. And then we also offer sequencing on platforms if you build your libraries yourself. So just a little bit about us. We are a very small team. There are only 8 of us. However, we produce a ton of data. As Molly said, we are pretty popular, and we've sequenced almost, I think, 2 petabases of data in the last 14 years. And to give you an idea of how big that is, one human genome is approximately 3 gigabases of data, and so this is about 1 million times that. So very briefly, we were established in 2001. This was the year that the first draft of the human genome came out. We've had a variety of names throughout the years, so you'll recognize us now as the SGT. We have sequenced, I have actually put up the wrong slideshow. I have a variety of, let me get the correct one. I have a number of iterations of our introduction. Okay, can you see the correct one again?
Yes.
Okay, awesome. Okay, so we left off, we were established the year that the human genome was sequenced. Since then, we've prepared and sequenced over 150,000 samples. Within the last 3 years, we've had 145 publications, many of those in Cell Science and Nature. And right now we're averaging about a $4 million revenue. One thing that's really important about us and that I'm particularly proud of is that we aren't just a run-of-the-mill genomics center. If you have difficult projects that you're working on or custom projects, that's actually where we excel. And I like to show this tweet from a scientist at the University of North Carolina in Charlotte, who gave us a shout out for working with her difficult sequencing samples, and that she thinks highly of our core, even though she is not at Duke herself. So we offer end-to-end services, so you can come to us and we'll give you a consultation about experimental design. We can extract your samples, prepare the libraries, which is the most difficult part, and then we can sequence your libraries, analyze that, and then we have secure data delivery. And as a researcher at Duke, you can come in at any one of these steps. So you can do the whole thing, or you can do something a la carte. So we like to stay really flexible. I do want to point out that we are a basic research core and we are not CLIA CAP. That means if you are developing a diagnostic for your research, we cannot develop that diagnostic for you. However, we can do anything up until diagnostic sequencing. And then I also just have the website in which you can check out everything. We have our services listed, as well as how to order. We try to make ourselves accessible to everyone. So if you need a consultation, there are links there. And I also should say that as a core facility at Duke Researcher, you get lower pricing than you would going to a commercial vendor. And then that is our basic introduction to us.